slide gives you a sense for how HIV processes its genome after replication. So here's the virus genome grouped into these three main categories, and you can see that in this particular slide here: GAG, POL, ENV. So this cluster of genes for envelope and polymerase and what's referred to as group antigen or capsid proteins. And so here's this representation of the messenger RNA. And imagine this is translated into proteins, which is in this particular cluster, the GAG polypeptide. In this particular cluster, the polymerase or enzyme polypeptide. And this particular case, the envelope proteins. And so the virus, in this particular case, produces this long polyprotein that has to be processed into individual capsid proteins and individual enzymes that it will carry with it, and then individual features of the spike protein. And it uses its own protease to cleave these polyproteins into these fragments so that they can then be folded up and mature. And so this slide gives you a bit of a overview of the entire replication cycle from initial attachment and fusion all the way to budding and exit from the cell. And so in this particular case, the virus GP120 docks on to its receptor, which triggers a membrane fusion event because these are enveloped viruses, releasing the capsid into the cytoplasm. The capsid breaks down, and the viral RT, reverse transcriptase, can then begin the production of minus strand and then plus strand DNA from the RNA template. Notice that the RNA is gone because the reverse transcriptase not only makes the DNA, the double-stranded DNA copy, but it simultaneously chops up the RNA while it does so. The virus also has proteins that allow it to enter the nuclear pores and then integrate as a prophage into the nucleus. In this prophage state, the virus can then be transcribed into messenger RNAs, which will serve as the genome. Some will serve as the genome, and some will be exported to be translated into these polyproteins that I talked about. So here's different length polyproteins that then get chopped up into their individual pieces, and they can associate with the virus particle in order to make a complete virus particle full of the associated enzymes that it needs. In this particular case, the integrase, reverse transcriptase, and the protease. And then you have the nucleocapsid proteins, the capsid proteins, and then the matrix proteins that give this guy structure. Some of these transcripts move through the ER, and those are the envelope proteins, the spike proteins, GP120 and GP41. And those are trafficked through the Golgi in vesicles and then set up shop in the membrane, and that serves as a focal point for the matrix proteins and so forth to assemble. Eventually, this leads to budding, and the virus releases into the world. And so in this particular cartoon, or animation rather, you can get a sense for that, where the virus is enveloped. Here's its surface proteins. Here's the transmembrane protein. Here's the capsid, this conical capsid, the matrix proteins, the RNA, the diploid RNA. Here's the RT, that little red sphere, the protease, the blue sphere, and the integrase, the purple sphere. So this virus then docks on to its receptor. GP120 finds its receptor, the CD4 antigen receptor. There's the CD4. There's the co-receptor called CXCR4, and or CXCR5, and that binding event facilitates the membrane fusion, and that facilitates in turn the release of the viral genome and enzymes into the cytoplasm. The reverse transcriptase then, you'll notice, makes a single copy of the DNA, then the double copy, and then the double strand. The RNA is vaporized, and then the DNA circularizes and then can enter the nuclear pores and insert as a prophage using the enzyme integrase that the virus brings with it. So there's our proviral or prophage DNA inserted into the host chromosome. Now the host polymerases can transcribe this. So there's a host RNA polymerase making messenger RNA from the DNA template. And so some of the RNA is used as genomes, and some of it is used. Some of it's used as genomes, and some of it is used to translated into proteins. And so the protease comes over and matures those proteins, and then those proteins become pieces of virus that can then be inserted into the membrane. So there's the spike proteins, the GP120, GP41, and then the rest of it, the matrix proteins, capsid proteins, the viral enzymes, and more genome pieces. And then the virus buds out into the world.